In an age where our society has become so sophisticated, why do so many elderly individuals not remember some of the most important information about themselves and their family members? The simple answer is tau protein and its effects on the progression of Alzheimer's disease. So what exactly is a tau protein? Tau proteins are proteins that perform the functions of stabilizing and providing flexibility for the structure of microtubules, which are found inside of a neuron on the distal portion of its axon. The exact location of the tau protein can be located over 100 kb on chromosome 17, band position 17q21, as seen on the picture to the right. Tau proteins stabilize microtubules through their 6 different isoforms and the process of phosphorylation. Phosphorylation is the addition of a phosphoryl group to a molecule. These molecules are a cis and trans version of the tau protein isomer PT231-pro-tau. When you look at the trans isomer, you see that the microtubule assembly rate is considered high along with its dephosphorylation rate within the molecules. You can also see that the molecule's stability and aggregation are relatively low. This isomer can be associated with Alzheimer's disease. The trans isomer also has a low neurodegeneration rate and is mostly found in healthy neurons. As you can see, the cis version of the protein makes most of these variables act in the opposite way. This cis version has been linked to Alzheimer's disease and mild cognitive impairment. However, both of these isoforms are currently being used for the production of antibodies and vaccines in order to treat Alzheimer's disease as well as mild cognitive impairment. The malfunction of tau protein is caused by hyperphosphorylation. Hyperphosphorylation causes tau proteins to dissociate from microtubules, which causes the formation of neurofibrillary tangles.
How are neurofibrillary tangles formed? When a neuron wants to transfer its signal to another neuron, it starts from the soma and transfers a signal to the synapse. Signal passes through the skeleton of the neuron, which are called microtubules. These microtubules are stabilized and kept in place by tau proteins. Alzheimer's is thought to be caused by defective tau proteins that unbind from the microtubules, causing the microtubules to dissociate from themselves, making it unable to maintain its structure. The defective tau proteins then turn into filaments that assemble in the neuron. Without the skeleton, the neurons lose connections between each other, thus causing them to become neurofibrillary tangles, which eventually causes the neuron to die. Tau proteins were discovered by Mark Kirschner and his team at Princeton University in 1975. This led way to a better understanding of the pathology of Alzheimer's disease. Currently, there is no known cure for this disease. However, we now better understand the progression of Alzheimer's disease and what might cause it. Hopefully, with the progression of modern medicine and what the scientific community has already learned about tau protein and its effect on the progression of Alzheimer's disease, we can find a way to treat or even cure the disease in the not so distant future.